Okay, so thus far in this class, we've discussed amino acids, we've discussed protein structure, we've discussed how the amino acid sequence can direct three-dimensional structure, right? So secondary structures such as alpha helices, beta sheets, right? Tertiary structures, how those multiple secondary structures create the three-dimensional form of a single protein, and quaternary structure, how multiple proteins might come together to create a macromolecular complex. So we've talked about structure. Now we want to talk a little bit about how structure of proteins then relate to their actual function. Now, some structural motifs that we've talked about lead to the function of traversing the membrane. And here we're going to talk about how that structure might regulate enzymatic activity. And we're going to do that in the context of this disease known as botulism. Okay. All right. So botulism is a disease caused by a bacterial infection. The bacteria is Clostridium botulinum. It is a spore-forming bacteria that's really common in the soil. So, of course, being the parent of recent toddlers, you very routinely teach your kid, do not eat dirt. This is maybe one reason. So how then do adults and kids that have been taught not to eat dirt, how do they wind up getting botulism? So one way is improper storage of food. There is another way. Does anyone want to fathom a natural way? Yes. Absolutely. So there's a way that there's something that we purposefully ingest that can give you this. Yes. Yeah, so certainly washing, food preparation is a part of it. What's something that you might intentionally ingest? Does anyone know? It is, but I'm not purposefully ingesting that. But you're right. At least not today, I'm not. Yeah. Yeah, but again, that's really about a food preparation. So honey. So botulinum toxin is actually in honey. So you do not give your babies honey when they're little. Of course, you'd have to ingest so much honey, you'd get a sick stomach before you would develop botulinum, have the effects of the neurotoxin, but it is actually in honey. All right, so what are the symptoms? Flaccid paralysis sounds awful, and it sort of is. This is where you lose the ability to contract muscles. All of your muscle contraction ceases. This obviously makes it pretty difficult to swallow and speak. You can see clear signs of facial weakness here in this gentleman here on the right. Droopy eyelids, right? Dyspnea. This is probably one of the worst. Anyone know what dyspnea is? Yeah. Exactly. Trouble breathing, right? So my diaphragm is what is actually causing a negative pressure on my lungs and allowing me to inhale. So if I cannot contract my muscles, I can't contract my diaphragm, I can't pull a negative pressure, and I can't breathe. Okay? Breathing, sort of important for life, so not really great. All right. But before you die, you will suffer from nausea, vomiting, and other things. All right. So, the prevalence of this disease is quite rare, right? We're actually pretty good about washing our fruits and vegetables, thankfully. And apparently our kids do listen to at least one of our lessons, which is don't eat dirt. Okay. Botulism works via the secretion of a toxin known as the botulinum toxin. Botulinum toxin is an enzyme, which is why we chose this disease. Okay. Yes, botulinum toxin is Botox. And you can imagine if I got my little crow's feet at age, I won't tell you, and if I want to remove them, one way I can do that is to relax all the muscles in my face, and so I can get shots of Botox in my face and relieve some of the lines that you clearly do not see from way up there. All right. It is among the world's most deadly toxins. The LD50 is about 100 nanograms for an average person. Anyone want to fathom a guess what a LD50 means? Yeah. What does that mean, median lethal dose? Yeah, so you're really close, and I just want to be really specific. Yeah, go ahead. Yeah, that's the specific definition of a lethal dose. LD50 is if I give 100 people this particular dose, 50 of those people will die. Okay. All right, again, this toxin is an enzyme. Everyone saw the pre-lecture. An enzyme is a protein that catalyzes a reaction, right? Facilitates a reaction. All right, so we're very briefly going to review some of the material that was in the pre-lecture, and that's the Michaelis-Menten model for enzyme kinetics. Again, this is I'll reiterate what Dr. James said in the pre-lecture, which is that this is a model. This is not a law of enzyme kinetics. It's a model to predict and to estimate what the conversion of substrate to product under the conditions of that specific enzyme. Vmax, remember, is the rate at saturation for a given enzyme concentration in moles per unit time. Please be really specific when you're studying about like how this is worded. The rate at saturation, right? It's an asymptote here when the substrate concentration is very high for a given enzyme concentration. That's critical in moles per unit time. All right. The Michaelis constant, or Km, is the substrate concentration that gives the half maximal velocity. These are oftentimes, well, always determined experimentally. So we described in the pre-lecture how you determine this, how you establish this curve. From that curve, you can't actually determine the precise Vmax, but you can find the estimate for it, right? We're going to go down half of that Vmax, and we're going to use that parabolic equation in order to derive the Km, or the Michaelis constant. Okay. All right, couple of assumptions that were harped on in the pre-lecture, and I'll harp on them once again, which is that the enzyme substrate complex is at steady state. Remember, it's E plus S, and there's a reversible reaction to the ES complex. That's simply the association constant, right? How quickly they associate and how quickly they dissociate. So if I were to write that up, right? E plus S. In the forward direction, we call it K1, but that's also known as the association constant. It gives rise to the ES complex. The reverse is K minus 1. That's also what we call the dissociation constant, right? So this is just a binding event, okay? All right, that has to be at steady state, which means that there is no net change. The delta per time of the enzyme substrate complex is 0. It's not changing its concentration, okay? The other is that there is way more substrate than there is enzyme, right? So the enzyme, if you remember from the equation, goes to E plus product. E is regenerated. It comes back here and continues to participate in the reaction. S is consumed, right? So what we don't want is S to become a limiting factor, because then all of the equations fail, okay? So the idea is that you can never get rid of the substrate, okay? All right. I can't harp on this enough. You guys just focus on this slide know this equation. It's not blue, but I'd strongly suggest that it be in blue. All right, so let's test our understanding of this. All right, here's a little problem for us to do in class. You do an enzyme rate experiment and get the following values of V at different substrate concentrations. Km is approximately what? All right, I'm going to give you between 60 seconds and 120 seconds to figure this out. This is your experimental data set. At this concentration of S, you've measured a velocity of this. Yes, S in a bracket. If you have brackets around, in this case, S, that would be a concentration. If it's around E, that would be the enzyme concentration. You can. I don't think you'll need to in this, but you certainly are more than welcome to. Anyone want to fathom a guess? Yeah. 1.1, 1.2. You want to work through the math for me? How did you estimate that?
We just kind of estimated for about like 5.6. So it goes to 12, like max velocity is around 12.
Okay.
So then half of that velocity is about 6.
Yep.
And then it looks like we see 5.6 and 9. And one's pretty close. One is the concentration at velocity 5.6.
Yeah. Does everyone understand that? So what he did was he estimated the Vmax. So the only way you can do that is that your velocity numbers, right, measurements, start to plateau. If they look like they're going linear, we haven't given you enough information, right? You have to see where your saturation is, right? This is the point of saturation. So he observed that the data go to saturation right around 12, 12.2 probably. He took half that, that's the Vmax, took half of that, it's around 6, 6.1, something like that. And he looked at the data set and said that, well, the half max is somewhere between the 5.6 value and the 9 value, which is somewhere between 1 and 2, and he made an estimation that's probably closer to 1 than it is to 2, right? So I'd say 1.1, 1.2, somewhere in that zone seems like a reasonable guess. Yes? Remember, when you're finding the Michaelis constant, the Michaelis constant is the concentration of the substrate that gives rise to the half max velocity. Right? So again, if I'm drawing the curve, here's Vmax. The Km, right, is here. This is substrate concentration. Km is the concentration that gives us half Vmax. That's the definition of the Michaelis constant. Okay? It does not equal 1/2 Kmax. It equals the substrate concentration that gives rise to the half max based on that enzyme's activity. No. So here, these are experimentally derived values, right? So someone gets a concentration of 0.5 nanomolar and measures this velocity, velocity 1, which is 2.7. They do 5.6 concentration. They measure, 1. and they measure 5.6 as a velocity. They do 2, they measure 4, they measure 8, and they measure 16. That's what they're doing. They're using the substrate concentration and they're calculating Vmax. If you need help on this, I would suggest that you go back and watch the pre-lecture where Dr. Janes goes through exactly how we go through measuring this crap, I think is how he puts it. Okay? All right. So let's keep moving on because we do have a lot more to get through today. All right. So think back to the pre-lecture. Here's another thought experiment. Which of the two of these illustrated reactions is going to go faster, A or B? Why? All right, I heard it here, and then I think I heard it reverberated. So the activation energy is lower, right? All right, so let's talk about inhibitors, and then we're going to talk about regulation. So obviously inhibitors, an inhibitor is a type of regulation which implies that we're going to downregulate. We're going to reduce the activity of enzymatic activity. And the reality is that many drugs like Cipro, a lot of the anti-HIV drugs are actually enzyme inhibitors. There are two main kinds of inhibitors that we're going to expect you guys to be responsible for knowing. One is competitive and one is non-competitive. As you can imagine, then competitive inhibitors are going to bind to the active site of the enzyme. So they're competing with substrate for the same site. Okay? So anyone want to remind me how we designate, how we label or call or name enzymes from the pre-lecture? They always end in something, yeah. ACE, that's right, A-S-E. So we're going to call this one triangle protease, all right? So it's going to bind blue triangle. It's a peptidase or a protease, so it's going to cleave that triangle. What a competitive inhibitor is going to do is it's going to bind that pocket, but it doesn't look like triangle. Not quite, right? It's got enough difference that the enzyme recognizes it, but can't actually facilitate that catalytic reaction, in this case, proteolysis. All right. These are going to alter the Michaelis constant, but not Vmax. Why? Anyone want to fathom a guess? Yeah. To Vmax, right? So Vmax is a characteristic of the enzyme, and we haven't changed the enzyme's activity. We're only binding up and blocking its active site, okay? So we're changing the concentration that's going to be required in order to reach the Km, right? All right. So what's going to happen if so one way to answer this as well is what will happen to V, to the velocity of this reaction, if we push the substrate to very high concentrations. What will happen? Yep. What's that? It'll max out. What does that imply when I tell you that nothing happens to Vmax, right? That we haven't altered Vmax. And I go to infinity substrate. Yeah, the enzyme still becomes saturated. Vmax is a property of the enzyme, so the inhibitor ultimately fails at very high concentrations, right? Because we're not actually altering the enzymatic activity, right? And so what you'll find is that if I were to draw this out, oh, that's kind of tailing off. All right, here's Vmax. When I add a competitive inhibitor, what I'm going to get is something that looks like that. It's still headed to the same Vmax, but now when we look at the half Vmax, I've increased the Km, right? So we'll call that Km prime versus Km. So I've increased the concentration required to get to that half Vmax. All right. Okay, non-competitive inhibitors are fundamentally different. Here, they're going to bind somewhere else besides, this is still blue triangle protease, but our inhibitor is going to bind somewhere else, a distant site, an allosteric site, and it's going to regulate then the enzymatic activity. Allostery, or the allosteric site, refers to a process known as allostery. Allostery is action from a distance. It's the easiest way to remember it. Action from a distance. So I'm a relatively large protein, and I can have something bind a distant site and affect the enzyme's ability to convert substrate into product. How might it do that? What's one unifying theme in our protein conversations? Yes? Yes, yes. It changes the structure, right? We induce a conformational change, which is going to disrupt that catalytic site or alter the catalytic site so it's less effective. So a theoretically perfect non-competitive inhibitor is only going to alter the Vmax. It actually won't affect Km. Got an eraser here. All right. So in that case of a non-competitive inhibitor, here's Vmax. Now, a non-competitive inhibitor is going to look something like this, right? So this is Vmax prime. So we've changed the Vmax. If I look at half of this guy, well, he's kind of inhibited it by 50%. So the half Vmax of this guy is right here, and there's his Km. And the half Vmax of this guy is somewhere right around here, and his Km is right there, right? So that's the theoretically perfect non-competitive inhibitor. You haven't changed the Michaelis constant, but you have changed the Vmax. All right, so here it is. It converts substrate into product. This is a non-competitive inhibitor. It binds at an allosteric site, and it changes the structure of that catalytic domain. And now triangle can't bind circle, right? Okay. So what will happen in this case to the velocity of the reaction if you push the substrate very, very high? I'm sorry, one. Correct. Yeah. Yeah. Well, so there are a number of reasons, right? So it's a great question. So let's say that I have a protein that is binding to the top side of this enzyme, and it's doing it through ionic interactions. Now I've changed the surface energy of that protein, right? And those ionic changes, let's say I actually, those ionic forces and ionic bonds are strong enough to pull or disform, right? Deform that protein. Because it's a single, in this case, a single protein, those changes usually are transmitted throughout the entire protein, right? And so it's going to be through ionic interactions, hydrogen bonds, all the same things that regulate protein structure are going to also, when two proteins come together, or in this case, an inhibitor binds an enzyme, those are the same forces that are going to drive conformational changes, right? Okay. All right. So when we talk about regulation, we've talked about inhibition. Inhibition is a form of regulation. Let's talk and expand this concept a little bit to allosteric regulation, which is when a regulatory molecule, it could be an inhibitor, but it could also be an enhancer, right? So in this case, we're going to bind to a site separate from the active site, and we're going to alter the shape, right? So we're going to change the conformation of the enzyme to change its activity. But when we talk about regulation, it can go both in the positive direction and the negative direction. It can actually increase the enzymatic activity in this way. All right, these enzymes that function this way usually have what we call a catalytic, right, or the active domain, and what we would call a regulatory domain. There's a little cartoon. Here's R, a lot of times denoted by R, the regulatory domain. C, the catalytic domain. Here's the allosteric site. You get this, in this case, an allosteric inhibitor, but it could be an allosteric activator that binds to that regulatory domain and changes the shape of the catalytic domain, in this case, such that it can't turn this substrate into these two products. Okay? All right, so I'm going to back up a little bit and just talk about some vocabulary, just so that we're really clear. And what I want to focus on here is the difference between what we call a domain and what we call a subunit. Domains are structurally and functionally distinct regions of a single protein. One long polypeptide chain that's formed a tertiary structure that has multiple, both structurally distinct and functionally distinct regions within that single protein. Okay? Here's an example. This is a protein, just a random protein pulled up, and you can see even just by eye. It doesn't take a rocket scientist to figure out this domain could be described very distinctly from this domain here. So domain A and domain B. I can't tell you what domain A and domain B are on this particular protein. It could be that this domain A is the catalytic domain and this is the regulatory domain, or vice versa. But the point is that they're both structurally and functionally distinct, but it's a single protein. The difference is when we talk about a subunit. A subunit implies that there are two separate proteins that have come together, right? And those two subunits work together, right? One may have a regulatory activity and the other one may have a catalytic activity, but they're two separate proteins that are working together. That's the key difference. All right. So if I'm talking about this type of structure, what type of structure is this here of a single protein? This is back to tertiary. What is this structure? Quaternary. What's a protein that we've talked about in the past already that is demonstrative of a protein with multiple subunits? Yeah. A ribosome is a pretty complicated example, but yes. What's one that we actually used? Hemoglobin. Not the red blood cell, but the hemoglobin within, right? Had two A subunits and two B subunits. Those are four distinct proteins that were coming together in this macromolecular complex to function. All right, if we look at the structure of botulinum toxin, we find it is a single protein, and it's divided into three domains: a receptor binding domain, a translocation domain, and a catalytic domain. Okay? I do want to make a note that chain is an ambiguous term. You're going to read papers and it's going to say this chain, that chain. Chain does not give you any information to imply whether it's a single protein or multiple proteins, okay? So just be careful about reading chain and assuming that means subunit or domain. It does not. It has to do with, it's a historical designation of a lot of times how the proteins were discovered. In this case, as you'll find out, this single protein as a part of its activity is cleaved right here in this domain, right, during its natural lifetime of this protein. And when you're isolating botulinum toxin, depending on the state that it's in, it appears on Western blot or by SDS gel as two bands. And they said, there's a heavy chain and a light chain, right? So it's a descriptor of an experimental process. So just be real careful, okay? All right. So different types of regulation. We've just talked about allosteric regulation. Another type of regulation is that of covalent modification. All right, here's enzyme X, and by adding or removing groups, right? In particular, we'll use this example like a phosphate group by kinases. Kinases are a specialized type of enzyme that catalyze the phosphorylation or the addition of phosphate groups onto specific amino acid residues. There are enzymes that remove those phosphate groups called phosphatases, right? So kinases are going to add those phosphate groups. Phosphatases are going to remove those phosphate groups. Kinases and phosphatases, the reason I'm kind of harping on it is that when we get to signal transduction, this is a major mode of signal transduction, how signals are propagated from the cell membrane all the way to the nucleus, or through phosphorylation and dephosphorylation enzymes. All right, so here's X enzyme X with X kinase. Let's say it is a kinase. Sorry about that. It's going to phosphorylate a residue, and as a consequence of the addition of all that negative charge, right, we're changing the energy landscape of that protein and now shifting ionic bonds around, perhaps disrupting hydrogen bonding. You're going to get a shape change, and as a function of that shape change, you're going to alter the activity of that enzyme. Just to remind you, the amino acids that are routinely phosphorylated on our proteins in our bodies are serines, threonines, and tyrosines. Again, if you're reading about signal transduction, tyrosine phosphorylation seems to be the most prevalently studied. It's unclear if it's the most prevalent mechanism in our cells, or a lot of times you'll see lots of literature when we have good reagents to measure. We have great reagents to measure phosphotyrosine. We have far worse reagents to measure phosphoserine and phosphothreonine. All right. Another way is to cleave bonds. So we can convert things like proenzymes. Remember I talked about this designation of proteins. Pro in front of a proenzyme or a protein implies that there's some type of processing step to get the mature product. In this case, it's an enzyme that has to undergo some type of, in this case, proteolytic cleavage in order to become active. We call these proenzymes. You'll also see them in literature called zymogens. The best example of this is the blood clotting cascade. If you go and look at it, it is a bunch of zymogens that activate one another in this long cascade that leads to the activation of thrombin, and thrombin cleaves fibrinogen, and it self-assembles into a polymer. And that's how we stop, one of the reasons why we stop bleeding. Okay? So enzymes that act on other enzymes cleave through proteolysis to activate or deactivate, right? So we're going to find that out. Botulinum toxin winds up being a protease that inactivates a key protein in our body. All right. Finally, another type of regulation is that of association and dissociation of subunits. Again, I'll use the demonstrative example of something like TGF-beta receptor, which is two subunits that are separated in the plasma membrane, and the binding of TGF-beta induces their association with one another. When those two halves of the TGF-beta receptor come together, they activate, they become active, and now can translate their signal. All right, they're actually known as a receptor tyrosine kinase. They're a receptor that has kinase activity in the tail domain, right, in the intracellular compartment. Okay. All right. The final that we're going to talk about is cooperativity. And again, this one's a little more complicated. I think maybe we're doing okay. Cooperativity is when a product, right, one of the products promotes the formation of more product. So I have an enzyme. It's going to act on a substrate and generate a product. The best example of that is if that enzyme has a binding site for that product, which changes its conformation and enhances its enzymatic activity. Yeah. Yes, exactly. Positive feedback loop. Now, it can also be feedback, but negative, right? So it could be that in many cases, the production of product actually comes back and inhibits the enzyme. And that's a way that we make sure the enzymes don't run amok, right? Is that their products begin to lower their activity. All right. In this case, it can imply multiple catalytic subdomains or other types of self-reinforcing catalysis. And in this case, we can't use the straight-up Michaelis-Menten model for enzyme mechanics. Enzyme kinetics. In this case, a gentleman by the name of Hill actually trying to figure out how on earth to explain oxygen association with none other than hemoglobin. Right? So hemoglobin, the 4 subunits, actually exhibit cooperativity, where the first oxygen is the hardest to bind. Once that oxygen binds to one subunit, it enhances the affinity of the other subunits to bind additional oxygens. So you can almost actually, it's very difficult to get it on. It's actually very difficult to get that last oxygen off as well. And you guys will learn more about that in physiology. So he described this factor known as the Hill coefficient, which is modifying the standard Michaelis-Menten equation in this way, where it becomes an exponent for the concentration of substrate and an exponent on the Michaelis constant. This is discovered or determined experimentally. And it can be a non-integer as well. It can be 1.5, 1.3. It is, we're measuring the kinetics of that enzyme, and we're almost reverse engineering the equation to model that behavior. Okay? But you can see where the Hill coefficient, here are these n's, a Hill coefficient of 1, 2, and 4 is indicating a degree of cooperativity within that enzyme. And I hope what you can see from these curves is that the higher level of cooperativity, the sharper that curve becomes. It goes from parabolic to sigmoidal, and the concentration range with which that enzyme now is operating becomes much tighter, right? So this is a really great way that we control when enzymes will become active and when not, right? Okay, a good example is that of glycogen synthase kinase 3. And get ready for this kind of designation. There's even a MAP kinase. And it has to do, again, with historically how things were discovered. Somebody discovered MAP kinase, and they're like, oh, wait, it's regulated by another kinase. Well, we've got to call that one MAP kinase. And then somebody else came along and said, oh, there's another kinase that activates that kinase, so that one's called MAP kinase. So you can see it tail spins really quickly. So in this case, glycogen synthase kinase is a kinase that is going to phosphorylate glycogen synthase. Glycogen synthase is its own enzyme that synthesizes glycogen. And glycogen synthase kinase, or GSK3, recognizes this motif. Okay, when I describe a motif, we're talking about a linear sequence, peptide sequence, that is recognized by a protein. Okay, in this case, it is serine XXX. X means it can be any amino acid. Serine XXX phosphoserine. So what happens in this case is another enzyme, another kinase called CK2, phosphorylates this first serine. GSK3 recognizes serine XXX phosphoserine and phosphorylates that serine residue. Of course, now it recognizes the next serine XXX phosphoserine and continues to phosphorylate down that chain. Okay? All right. Okay, so we have 15 minutes, I think, actually almost 20, so I think we're about on time. So is everyone able, actually it doesn't matter, I'm going to play this video for you on basic neurotransmission, and then we'll get into it. Oh, and it's not playing. Okay.
The action potential cannot cross the synaptic space, but it reaches the axon terminal. It causes membranous sacs called vesicles to move toward the membrane of the axon terminal. The membrane of the vesicle fuses with the membrane of the axon terminal, enabling the vesicle to release its contents into the synaptic space. The molecules released from the vesicles are chemicals called neurotransmitters. They drift across the synaptic space and bind to special proteins called receptors on the postsynaptic neuron. binding of the neurotransmitter to its receptor can trigger an action potential in the postsynaptic neuron. That electrical signal then moves toward the cell body of the postsynaptic neuron. Now that the neurotransmitter has relayed its message, it releases from the receptor into the synaptic space. Some of the neurotransmitter is degraded by enzymes in the synaptic space, and some of the neurotransmitter is carried back into the presynaptic neuron through transporter proteins. The neurotransmitter that is taken back up into the presynaptic neuron is then repackaged into vesicles that can be released the next time the action potential reaches the axon terminal. The entire process repeats each time an action potential reaches the axon terminal of the presynaptic neuron.
Okay, I think that's it. All right, so this is just a little bit of a primer. It's talked a little bit about membrane fusion, about diffusion of a soluble factor, about binding to receptors. So this is a great example of a lot of things we've already talked about. But what we're going to focus on in botulism and botulinum toxin is this process of vesicle fusion. All right, so we talked about membranes in the last lecture. The ability to get membranes to fuse is an extremely high energy effort. If there are any water molecules that are between the two adjacent phospholipid bilayers, you will not get fusion. And so we have a series of proteins that bind each other, undergo this, I don't want to say cooperative because we just use that term and it would be inappropriate here, but a sort of coordinated conformational change that squeeze all the water molecules out from between those two lipid bilayers and then allow fusion, okay? So that's what we're going to talk about. So botulism effectively impacts these two sort of diagrams here, it's a steered molecular dynamic simulation by this group in Denmark showing vesicular fusion and release of contents. So what does a synaptic vesicle then really look like? It's not these idealized cartoons that we tend to draw. It is a lipid bilayer that is absolutely chock-a-box full of proteins that do all sorts of things, right? One of those is this protein synaptobrevin, and it's the focus of the botulinum toxin. These proteins are going to do a number of different things, but in the case of vesicular fusion, the SNARE proteins, of which synaptobrevin is one of, are these proteins also known as SNAP receptors. And these are the proteins that are essentially labeling both the vesicle. The vesicle has certain vSNAREs, vesicular snares or vSNAREs, that give it its specific character, right? vSNAREs are going to bind to very specific T snares, or you can think about it as target membrane snares or target snares. The vSNARE and the T snare does two things. One is it helps to localize the vesicle to its proper location. So you can think about it almost like zip codes, right? My address is my T snare, and the zip code on the vesicle is the vSNARE, and only when they find each other will they induce a fusion event. Okay, so you have vesicles that are going all across the cell, and they need to fuse with certain domains or certain membranes and not others. Okay, so that's one, is that it helps target, right, the vesicle to the right membrane. The other is that when they bind each other, they undergo a conformational change that's coordinated with one another. So they look like this, they bind, and then they start wrapping around each other like a rope. So they form what we call a coil-coil structure, and it's just like a rope that you twist, right? If you hold one steady and you twist a rope and keep twisting that rope, all of a sudden it's going to start kinking and the length of that rope starts shortening, right? That's essentially what snares are going to do. All right? So synaptobrevin is the vSNARE that's found in synaptic vesicles containing the neurotransmitters. Here's synaptobrevin. It's going to bind syntaxin on the on the plasma membrane. It's going to undergo this sort of wrapping around one another, And as a consequence, it pulls those two membranes ever so close, squeezes out every single water molecule between the phospholipids, and then they can fuse, right? What botulinum toxin does is it's an enzyme that's going to cut synaptobrevin.